still it is very active, and we are currently analyzing in more detail what this protein does. So we then checked whether we can also reconstitute the second step of splicing, and we showed that only PRP16 and two second step splicing factors, SLU7, PRP18, are required to give an efficient second step catalysis here. And the way from further experimentation, I just summarize this now, how SLU7, PRP18 function is probably that they help position the 3' splice site of the intron into the second step catalytic center, and they help to dock the ends of the exon to a loop of U5 RNA, and they juxtapose now the 3' hydroxyl of the first exon, which now can ride a nucleophilic attack on the 3' splice site. So with this system in our hands, we could now apply also more sophisticated biophysical methods such as fluorescence cross-correlation spectroscopy, which would allow us now to monitor the dynamics of the coming and going of proteins along the catalytic phase. And the way we have designed the experiments is as follows. So we use a pre-messenger RNA which is coupled at the 5' end with an atto dye, and this is then incubated with an extract from a strain where we have expressed a protein of interest as a GFP fusion protein, so that we have a dual labeled activated complex, and we then add PRP2 in order to analyze now what PRP2 RNA helicase does to the activated spliceosome in order to get it catalytically activated. And I just summarize now many experiments, and what turned out that about 15 proteins of the activated complex get remodeled and in part lose their affinity for the spliceosome. For example, these proteins are displaced entirely from the spliceosome, and other proteins are now recruited, such as the step 1 splicing factor CW25. And particularly interesting are these U2 proteins. They are tightly bound to the branch site, to the RNA around the branch site in the activated complex. During the catalytic activation, these proteins get remodeled, and apparently they set this branch site adenosine free so that the 2' hydroxyl can now attack in a nucleophilic way the 5' splice site, and allowing this first step of the splicing reaction. We have also done similar experiments for the second step of the splicing, and I just summarize now what we think is the role of these step 1 and step 2 splicing factors, because what was known before is that there is only one catalytic center in the spliceosome, and that this can exist in two different conformations, two major states, which can be shifted by the PRP16 RNA helicase. And what CWC25 probably does by binding to the step 1 spliceosome, that it closes the catalytic center and now stabilizes the step 1 conformation, allowing for efficient step 1 catalysis. Then PRP16 opens up again the catalytic center, removes the branch site from the catalytic center, and now we have to get rid of the step 1 factor, and we have created by PRP16 a strong affinity binding site for SLU7 PRP18, and this now kicks out the first step factor and closes probably again the second step conformation, allowing now for an efficient second step. So we have also shown that these factors are not part of the catalytic center, so they are just modulating probably the conformation of the spliceosome. Now in the last few minutes I'd like to show you now, seeing is believing, some structures of the spliceosomes now switching to human spliceosomes, and here we are collaborating with Holger Stark, the cryo-EM group at our institute, and here we're using single particle electron microscopy, and this shows you now the structure of the pre-catalytic human B complex. These are the single particles. These are class averages here, which are used for the reconstruction of the 3D structure. It is about 40 nanometers long, and it has a thromboidal kind of structure. And the catalytically active C complex, so which comprises the step 1 intermediates, looks completely different. It has a major core structural element, but in addition we have lots of domains attached to this main structure here, a platform, a head structure, and you can barely recognize similarities between the pre-catalytic human B and the complex C. And this on the right-hand side is the dwarf, which is a 70S ribosome, just for size comparison with the spliceosome. And what we are doing now is we are using different approaches to learn more about the organization of these snapshots of the spliceosome, and it tells you you have to really analyze them one by one. And we do it in two ways. So on the one hand, we reconstruct larger subunits of some of the snapshots. For example, this is the trisnerp, and this is still an intact unit of the pre-catalytic B complex, and you can easily see the 3D structure can only be fit into this part of the B complex, and we have direct evidence by labeling studies that U2 and U1 is located here, as well as the pre-messenger RNA. For the C complex, this was more complicated. This is, by the way, the protein composition of about 100 proteins in the human C complex. And we happen to purify a large U5 RMP from nuclear extracts from HeLa cells. And interestingly, this shares the protein composition with part of the C complex and does not have U6 and U2 RNAs. And so therefore we reconstructed this 35S U5 RMP. This is the 3D structure, and trying to fit this into the native C complex. And the only way we can do this is down here in this size, and it fits in quite nicely, indicating that, and we have good evidence that this is the core U5 RMP, that this part is here in the native C complex. And from other studies, we know that U2 and U6 RNA are located in this area, so that we are very likely have the catalytic center sitting around here. And we have evidence for this now by direct labeling studies using an antisense oligo approach, which is biotinylated, and we can hybridize this to accessible regions of the intron or the exon. And we used 4 sites. The green area is shielded by spliceosomal proteins. And these sites, and then we use antibodies against biotin and label them with immunogold. And this is just one experiment localizing this region here upstream of the branch site. These are few examples from 100, 150 of these kind of single particles. We can, with confidence, localize the sites here at the surface of the spliceosome. These are the 2 intronic sites. These are the sites of the exon, and so we can tentatively roughly map the path of the pre-mRNA at the level of the spliceosome, and we would localize the catalytic center right here at this part, and this fits very nicely with our docking experiment with the 35S complex here. And with this, I would like to finish. So what we are driving at now is to go for higher resolution structures using cryo-EM, and we have also set up the pipeline to get milligram amounts of individual snapshots of the spliceosome and can start now crystallization attempts. On the other hand, we have also established a high throughput in vitro splicing assay, which allows us now to screen for possible splicing inhibitors, screening chemical libraries, and here we have teamed up with Herbert Waldmann, and in the next couple of months we will screen his library of about 200,000 compounds with the hope that we might get inhibitors of the splicing machinery and get more additional stable snapshots, which may hopefully in the next couple of years tend to crystallize. With this, I would like to finish and thank the people involved in this work, and in particular thank the collaborators, Holger Stark's group, for the 3D reconstructions done by Monika and Björn. We have a long-standing, very enthusiastic collaboration with Markus Wahl's group. We have seen one example here done by Karine and Zina on the BER2 crystal structure. We have collaborated with Jörg Enderlein at the Göttingen University. Thomas Ort is the driving force in my lab, a postdoc who is doing the FCCS experiments. Bertolt and Elmar have done the immunolabeling studies. Patricia and Zbyszek, they have reconstituted or established the reconstitution system, and Sergei has been involved in the purification of the human spliceosomes. And all this could not have happened with the extremely generous support by the Max Planck Society, and I think this is the only place in the world where I can have and can do these kind of studies, going for high-risk, long-term goals, and I hope yeah, we will get this money for another couple of years. Thanks very much for your attention.
Thank you very much, Reinhard. We have time for a very quick, sharp question. Please. Oh, this one?
Amazing question. I have amazing talk. I have a question. So you use a model RNA to work out how the spliceosome is assembled. Do you think the picture will change if you use different pre-messenger RNAs where the splice site sequences vary? And this is related to the fact that when you knock down some core components like SMB or U1C, you see more effects on alternative splicing than on constitutive splicing. So I wonder whether the picture of what you see will change.
It would take 5 minutes to answer all of the questions. So if we take different pre-messenger RNAs, both in yeast or the human system, the assembly pathway is more or less the same, which does not mean that in real life, let's say in the co-transcriptional mode of spliceosome assembly, that you may have also bypasses there. And it is a very flexible machine, so you can also reverse certain steps, which is probably very important for the fidelity of alternative splicing. And if we knock down certain factors, SNRP-specific ones, then you will also have even constitutive splicing factors, and then you go for genome-wide, let's say, alternative splicing readouts. Then you see very specific regulatory events on certain alternative splicing events. So it's not only that the bona fide regulators of alternative splicing are important, but also constitutive factors.
Thank you very much again.